Am I audible? Hello, can you hear me?
Yes, sir. Yes, sir.
Okay. So can you see the slides?
Yes.
Okay. So good afternoon to Anujji, and
Good afternoon.
a very good afternoon to all the members of this university. And I think is the elucidation of root and shoot specific molecular insight into phosphate starvation response in Arabidopsis. So first, if we can see the evolution of life, we can see the evolution of life. There is a 4.6 billion years ago, everything comes into the existence. The sun ignites, and then you can see gradually 4 billion years, 3 billion years, and 2 billion years, and then 1 billion years. The first prokaryotes came into the existence in 4 billion years. The first origin of life. And then, since then, life is start thriving, you can see. And then it's come to the end. The primates comes like 0.01 as compared, if we compare the overall picture. And then human Homo sapiens come to the existence. So 0.001. So we are very new. The one thing we always should understand that we are very new on this planet. And the writing skills, like the intellectual and everything, these civilization is just existed 0.0001. So we are just in a very new era. It's just happening. So if we introduce, there is two aspects of plants: nutrients and how plants adapt to the surrounding environment, how plants adapt to the surrounding nutrient condition. Here I'm giving the example of phosphate. Phosphate is the prominent energy source for the plants, for the photosynthesis, for the energy dependent reactions, for the oxidative phosphorylation, for the formation or synthesis of DNA, RNA, for the ATP. Everything needs phosphorus. And it comes from the non-renewable source of fertilizers, which is non-renewable. It will end in some day. It comes from the rock phosphate, and it's processed by different companies, manufacturer, fertilizer companies, and then they supply it in the market. So what happened when phosphate deficiency occurred to the plants? One most characteristic phenotype of the phosphate deficiency is formation of anthocyanin, induction of anthocyanin. Anthocyanin is a very important compound which has the anti-herbivory activity. It's a anti-aging activity. It is a common antioxidant. It's a very strong antioxidant. In wine, there is a red wine. There is a flavonoid, resveratrol, which is recently cited as anti-aging. So if you take the resveratrol, it gives you more younger life. Anti-aging, gives anti-aging effect. So this anthocyanin is very important for the human health compound. It's good for heart, cardiovascular diseases. It's a preventive thing. Whatever the color you can see around the world, color in the fruits, colored leaf, colored seeds, everywhere you can see the anthocyanin. Common anthocyanins are cyanidin, delphinidin, pelargonidin. These are the common anthocyanins. So you can see that, right? What they do, they are the anti-herbivoric, anti-osteoporotic activity, and anti-insecticidal activities. So they keep plants healthy. So if they keep plants healthy, they also keep human healthy also because of the same activity. There is a lot of research is needed how they functions in human health. There is a gap in the area, knowledge gap. We do not completely know how they works in human, what kind of metabolism they induce, what kind of signaling they induce to give health effect. But certainly, the one thing is, the bottom line is that whenever we take anthocyanin based compounds, they detoxify the body. The human, whenever they took anthocyanin based compounds, diets, anthocyanin based diet, anthocyanin rich diet, fruits, it will detoxify bodies by frequently people will urinate, and it clears the toxins from the body. It is the bottom line thing. And it's also the anti-osteoporotic activity in these compounds. Certain flavonols. There is a category, actually, the broad category is the flavonoids. And then the flavonoids, there is a subcategory is flavonols and anthocyanin. Flavonols are colorless, but this, very important, this was the they are considered as phytoestrogens. Phytoestrogen, which is analogous to estrogen and gives good bone health to the women. So I can show the general pathway of the flavonoids. You can see it all comes from the phenylalanine, the amino acid, and from the phenylalanine, different enzymes cascade comes and it makes the quercetin, camphorol. These are the flavonols, gives the rutin, and from this F3H enzyme, flavonone 3 hydroxylase, this branch turns into the anthocyanin. This is anthocyanin branch, and this is the flavonol branch. And there is other branch comes from this same phenylalanine amino acid is lignin. So the mother pathway or the pathway is phenylproponide pathway. Everything comes from the phenylproponide pathway. Also, if these flavonols or flavonoids or anthocyanin has the antiviral activity also. So role of phosphate, how phosphate creates induction of the anthocyanin formation in plants under deficient condition. So there is signal transduction, phosphate does, phosphate induces growth and development. As I earlier told, the phosphate is the structural component of nucleic acid, phospholipids, and ATP. And phosphate is the essential micronutrient, which made up of up to 0.3% of the plant's diet. Also, phosphorus governs all the energy dependent reactions or electron transport system. Am I audible? Everything is fine?
Yes, sir.
Hello?
Yes, sir. Audible.
Okay. So let me know if any interruption come or anything is hangs. Let me know, please. Thank you. So what next? How can we address the problem of phosphate deficiency? Because it is connected to the pathway of flavonoids, anthocyanin, which is very important in this era, in this pandemic situation. So we need to taking care of the phosphate deficiency. How should we address the problem? So there is a uptake the knowledge of phosphate starvation response. We need to update the knowledge. In the present study, we updated the phosphate starvation response. So what we did in our work, we used the STRA exon chip having 28,410 transcript cluster to map the phosphate deficiency response. How can we address the problem? If we increase the phosphate use efficiency in the plants by using the molecular information and modern biotechnological tools, then we can rectify or mitigate the phosphate deficiency problem. I mean, what I mean to increase phosphate use efficiency is you will put less phosphate fertilizer in the field, but plant will utilize it more efficiently. So there will be less need of phosphate fertilizer. What we did in our experiment, we used the hydroponic system to create the artificial deficiency of the phosphate in the media. Media is hydroponic, water-based media. In this modified MS media, we just remove the phosphate nutrients and everything remains the same. And we gave the treatment for 7 days treatment of phosphate deficiency. These are 5 days old plants. grown in the normal media. Thereafter, we transferred into the phosphate deficient nutrient media for 7 days. And then we sampled them root and shoot separately. And we produced, we extracted the total RNA from these plants. And we used further experiments like microarray and real-time PCR for validation of the gene expression. Before doing that, we in silico did the ionic speciation analysis of the nutrients. So what will happen when we remove the phosphate from the media? So our ionic speciation analysis shows that as soon as you remove the phosphate, the free activity of iron ions was increased. So 28.68%. And in the minus phosphate media, it is increased 37.49%. There is an increase in the free activity of iron. Same thing with the manganese, 55.8% free activity was manganese increased, and it become like 58.04%. Removing the phosphate from the media, the cation activity of iron and manganese was increased. It shows by the in silico analysis of the media. Okay. Then we did the total transcriptome analysis using the ST-array Affymetrix technology. And we found that there is a clear difference between the root and shoot under the same treatment condition. In minus phosphate, the root, this shows the root transcriptome. This triangle shows the root transcriptome, and this square shows the rectangular shows the shoot transcriptome. And these are far apart, showing the very much difference. They are very unique in their expression under the phosphate deficient condition. Second, the total number of gene is uniquely expressed in root under phosphate deficiency condition is 1,478, and in shoot is 2,591, and there is 1,012 gene is common between the root and shoot. So this phosphate deficiency thing is also studied or investigated by earlier reporters. There is a 10 to 12 paper which is completely specifically studied the phosphate deficiency response, and we also studied the phosphate deficiency response in our study. So how our study will unique? How can we differentiate our study from their study? So what we did, we subtracted all the reported earlier gene from our study to make our data novel. For example, in the root specific transcript, only 242 genes was earlier reported from different studies, and there is 1,236 genes were novel. Similarly, in the shoot specific transcript, there is 623 genes was reported, and there is 1,968 genes were unreported, unique. So it implies that our overall our data is very unique, very novel in this aspect because of the experiment and technology what we have used in this study makes it novel. The experiments, the hydroponic experiment is the novel thing because our hydroponic experiment did not have the agar-based media, because agar-based media itself has phosphate contaminated, because agar has the phosphate, different levels of the phosphate nutrients. And second is the ST-array. ST-array has the large number of probe sets as compared to ATH1 array. ST-array has 28,000, more than 28,000 probe sets. Then we validated our array expression data using the real-time PCR. Now come to these root specific events. Hello.
Devis sir, sorry for interruption. Please summarize your lecture.
Okay.
Please.
So if we see the metabolism, overview of the metabolism, the root specific metabolism, we can see the induction of secondary metabolites. There is some induction in the secondary metabolism. There is a heavy modulation in the cell wall and lipid metabolism. These are the cell function overview, root specific events. The regulation. So in the root specific things, the Virki transcription factor was highly upregulated. MIB transcription factor was modulated. There is a very heavy upregulation in the ubiquitin dependent pathway. And it's all come to the biotic stress, linked with the biotic stress. Jasmonic acid signaling is upregulated. And we found using the centrimotol, the root specific cis-regulatory element, we found all the Virki related elements. In the suit specific events, the photosynthesis, if we see the photosynthesis is totally downregulated. And the MIB related transcription factor is mostly upregulated, which controls the flavonoids pathway. Secondary metabolism, phenylpropionate pathway, we can see upregulated. And terpenoids pathway was also upregulated. And it's also related to the biotic stress. In the cis-regulating elements, there are BZIP and ethylene responsive factor was involved. And come to the conclusion of this study. We have summarized the model in which we have shown that there is no phosphate. It creates the nutrient imbalance, and it creates the oxidative stress. And this oxidative stress is upregulated the calcium ion channels, different calcium related genes, which upregulates receptor kinases and serine threonine kinases, and it upregulates protein modification, protein degradation, and it's modulated transcription factor, Virki-based transcription factor, which modulates genome-wide adaptive response. Overall genome-wide adaptive response leads to induction of flavonoids, glucosinolates in root, and there is hormone signaling. Ethylene is downregulated, jasmonic acid upregulated, and in the suit, the same mechanism, same signaling thing is different, which is upregulated in the root. We can see green is showing the upregulation and the black is showing the modulation. There is mixed up and down both. There is a contrasting response in shoot relative to root. The things which is upregulated in root, here it is either modulated or downregulated. Come to the transcription factor, there is MIB and BZIP transcription factor is specifically upregulated. And it's overall modulate the genome-wide adaptive response, which further photosynthesis is completely downregulated, completely shut down. Sucrose starch metabolism is upregulated. Oxygen signaling in shoot is downregulated, and again the jasmonic acid is upregulated. And these are finally leads to the modulation in the secondary metabolism, in which the flavonoids, terpenoids, lignins, glucosinolates, simple phenols all are upregulated, and the carotenoids is downregulated. This paper was published in the Environmental Experimental Botany, published by Elsevier. Acknowledgements.